In this work, we present netNorm technique introduced in the paper Estimation of Connectional Brain Templates Using Selective Multiview Network Normalization, published in the Journal of Medical Image Analysis in 2020. This work was developed at Basira Lab by myself, Salma Dhifallah, and supervised by Dr. Islem Rekik. Technological advances in the field of medical imaging has made it easier to acquire multi-view connectional datasets for large population of subjects. The wealth of connectional data can be a powerful tool for better understanding of neurodegenerative or neurodevelopmental disorders using group comparative studies. Yet, given the diversity of such complex datasets, it's challenging to identify a shared connectional mapping across subjects and across views, which represents the fingerprint of the given population. In this study, we propose a new methodology to estimate a unified connectional template for a population of multi-view brain networks. In other words, a population of different brain network views can be represented by a single brain connectional template called CBT. The question that arises at this point is, how can we integrate the complementary information offered by different modalities while reducing inter-subject variability between different subjects within the same population? In fact, inter-subject variability is the variation in connectional traits between subjects in a given population, let's say a healthy one. In such case, the estimated CBT should be robust against these variations, while spotting pathological connections in disordered brain networks as deviation from the normalized healthy template. Therefore, the estimated template has to be well representative for a given population by occupying a centroid position with respect to all subjects and all views. Also, it has to be discriminative in terms of brain connections that differentiate between different groups by well capturing population-specific traits. Now, I will give you an overview of previous works that tried to partially solve this problem. Rekik and co-authors presented in 2017 diffusive shrinking graphs technique, which uses iterative local fusion processes to estimate brain network atlases, which we also refer to in this work as connectional brain templates. Yet, this technique is only applicable to populations of unimodal brain networks or a single brain view derived from a single MRI modality. Dhifallah et al. Presented a clustering-based multi-view network fusion technique. First, the multi-view networks are mapped using nonlinear fusion for each subject in the population. Second, the fused views are clustered into different groups presenting similar connectional traits. By averaging each cluster, a representative network atlas is defined for each group. Then, the final CBT is defined as the average of the previously defined presentations. Despite its significant results on morphological data, the performances of this method depend on the number of clusters chosen in the linear fusion step. This parameter differs according to the dataset used. To address these limitations, we propose a solution inspired from Eric Fromm's social theory, which was introduced in the mid-90s. He assumes that, at the group scale, psychological traits are no longer defined by the complete image of an individual psyche. Rather, it is based on the common psychological features across the group members. Given a population of multi-view brain networks, the first step consists on extracting a feature vector using a pair of pairwise ing for each subject in the population, defined using pairwise connectivities across different views. Using the previously defined feature vectors, we construct a high-order graph depicting the interdistances between all feature vectors. Using this high-order graph, we model the relationship between different feature samples using Euclidean distance for a given pair of pairwise ing. As our goal is to define a CBT whose connectional features are close to all subjects in the population, we select the sample whose feature vector is closest to all other samples by satisfying the commonality criterion defined by having the minimum mean distance to all other vectors. As we repeat the previous steps for each pair of pairwise, we define a set of pairwise selected feature vectors or samples, which are then used to build a population representative tensor in a mosaic way, piece by piece. Therefore, the constructed tensor only contains the most common samples across the population, ensuring centeredness and representativeness. In the ultimate step, we define the final CBT by non-linearly fusing the different views of the population representative tensor using similarity network fusion technique (SNF) introduced by Wang et al. In 2014. To evaluate netNorm, we used a large dataset containing a population of 155 autistic subjects and a population of 186 normal controls. A small dataset was also used for validation, containing a population of subjects with mild cognitive impairment and a population of subjects with Alzheimer disease. Each dataset contains 4 morphological brain modalities presented by both brain hemispheres. We constructed each morphological view using a specific morphological attribute, such as sulcal depth, cortical thickness, or maximum curvature. For each cortical attribute, we derived a morphological brain network where the strength of connectivity between two brain regions of interest, referred to as airwise, denotes their dissimilarity in morphology. To evaluate the performance of netNorm in terms of representativeness, we calculated the mean Frobenius distance between the estimated template and each brain network in the population across all subjects and all views. We first used the whole dataset for evaluation. Then, and in order to ensure the generalizability of our proposed method, we used 5-fold cross-validation, where we divided each population into 5 subpopulations. We generated a CBT for each subpopulation and calculated its mean Frobenius distance to all views in the subgroup. The normalized Frobenius distance for each fold is calculated as follows. We take the Frobenius distance df and we subtract the mean Frobenius distance for each fold I. Normalized by the maximum minus the mean for each fold, we finally add a scaling term. Results show the effectiveness of netNorm compared to state-of-the-art methods by occupying the minimum distance for each brain hemisphere for both ASD and NC populations, as well as LMCI and AD groups. Now moving to the discriminability evaluation of netNorm in comparison to state-of-the-art methods, where we first identified the most discriminative regions of interest in the brain in distinguishing between two groups of subjects using the estimated template in comparison with multiple kernel learning (MKL) technique. Then, we calculated the matching rate between top discriminative airwise identified using MKL and using the estimated template for netNorm and state-of-the-art methods. For multiple kernel learning technique, we started by linearizing the upper triangular part for each brain network, defining connectivity features for each network in both populations. Using the labeled feature vectors, we associated a kernel for each element. Then, we traced an SVM classifier that outputs a weight vector denoting the discriminability of each feature in the classification process. In the next step, we defined a score vector that assigns a score for each RY as a sum of the weights of all connectional features related to it. Top discriminative RYs are then identified as those having the maximum score. While using the estimated CBTs, we simply started by applying the absolute element-wide difference in terms of connectivity variation between both CBTs. By summing up the columns of the difference matrix, we get a score value for each RY. Those with the maximum scores are selected as the top discriminative regions differentiating between healthy and disordered populations. We applied these steps using CBTs derived from both netNorm and state-of-the-art methods. Results show that netNorm had the best overall matching rate in discriminative airwise identified using the estimated CBT in comparison to a completely different machine learning method, which is MKL technique. This demonstrates the reproducibility of our results in terms of the identified discriminative regions revealed by our CBT. In this table, we display top discriminative airwise distinguishing between healthy and autistic subjects using the left hemisphere. Top regions include pericalcarine cortex, which presents thicker cortex in autistic children according to literature. Internal cortex, responsible for hyperactivity, aggression, and self-injurious behavior in autistic subjects. Isthmus cingulate cortex, causing social behavior impairments. Pars opercularis and pars triangularis, affecting social communication and language respectively. The discriminative airways for the right hemisphere also included pericalcarine cortex and internal cortex, in addition to the rostral interior cingulate cortex, responsible for aggressive behavior in autistic subjects. Fusiform gyrus, causing difficulties in face perception and person's recognition, and parahippocampal gyrus, causing abnormal social cognition function. In conclusion, in this work we present netNorm technique, a framework used for unifying a population of multi-view brain networks using a selective fusion process. Results show that connectional brain templates produced using netNorm are centered and well representative for a given population, in addition to being discriminative in terms of preserving the population traits. In future work, we tend to apply manifold learning techniques in order to enhance the nonlinear fusion step, in addition to evaluating netNorm on other types of brain network views, for example, functional brain connectivity.